Good day, class. So for this meeting, we're going to tackle the second to the last lesson for the midterms, your introduction to virology. So we are done with your mycology class. This time, we're going to discuss a brief overview or a brief background on what is virology, what are the viruses involved, and the lab tests that would be done for you to help in the diagnosis of different virus diseases caused by viruses. Let's start. Now, let's have a quick history class regarding your virology. Now, in the late 1800s, scientists Adolf Mayer, Dmitry Ivanovsky, and Martinus Beijerinck discovered the first virus class known as your TMB, the tobacco mosaic virus. Now, itong tobacco mosaic virus na to, kindly take note, was isolated from tobacco, yung ginagawang sigarilyo class sa probinsya. It was isolated. They were able to isolate it from tobacco, this tobacco mosaic virus. Now in 1898, the disease foot and mouth or the foot and mouth disease was discovered. And from their class, the first filterable agent from animals was discovered. So the first filterable agent from animals were discovered. During that time kasi class, iniisip nila, they were thinking that foot and mouth disease was caused by a bacteria. So what they did was they used porcelain porcelain filters. And from that porcelain filters class, di ba ang mga porcelain nyo, if you view it microscopically, yung porcelain, may mga maliliit na pores yan. Now, what happened is that yung virus that would cause foot and mouth disease was attached to the pores in the porcelain filter, which became the first filterable agent. Now, this was first described by Loeffler and Frosch, and this was the start of the discovery of the first filterable agent. Then in 1901, the yellow fever virus was discovered. So yung yellow fever virus is known to cause your yellow fever. This is the first human filterable agent. Take note of that. Yan yung first human filterable agent. Yung first filterable agent na discovered was the virus causing foot and mouth disease. Pero yung first human filterable agent is yellow fever virus. This was discovered by Walter Reed. Now, during that time class, they were able to use the word virus. Now, virus is derived from the Latin word for slimy liquid or poison and was gradually introduced to replace the term filterable agent. Now, take note class, yung TMB, yung tobacco mosaic virus, was the first virus visualized using X-ray crystallography in 1941 and electron microscopy in 1939. Then in 1915, Frederick Twort and Felix de Herel were credited with the discovery of viruses which could infect and lyse bacteria. So they were able to discover what we call bacteriophages class. Itong bacteriophages na to, these are viruses that would target bacteria. De Herel introduced the term bacteriophages for agents capable of virus absorption to its target, followed by cell lysis and release of infectious material on bacteria. So, kunwari class ito yung bacteria niya. What happens is that your virus would attach, would absorb. Absorption is attachment class. Mag-a-attach siya sa surface niya. Mag-a-attach siya sa surface niya. Then, it would penetrate or it would destroy the cell wall. Then, it would copy or it would get the genetic or the nuclear material, yung DNA or yung RNA ng bacteria, and it would use it to create more bacteriophages. Then in 1949, the tissue culture system was discovered. Now, ang viruses nyo kasi class, please take note, viruses cannot be grown in agar. Now, in 1949, the tissue culture system was discovered. So instead of using agar, they use tissues. It could be animal tissues and animal tissues class. Yun ang mostly ginagamit dyan. A major breakthrough that led to the isolation of poliovirus. This was discovered by Enders and his colleagues. Breakthroughs would include development of the poliovirus vaccine, discovery of new agents and diseases, more understanding of molecular biology and cancer. Now let's go to the definition of terms class. So let's start with your virus particle or your virion. Now when you say virus particle or virion, this is an infectious agent composed of either RNA or DNA. If you still remember in your mol bio, if it's RNA or DNA, they are both known as nucleic acid. So your nucleic acid could be RNA or it could be DNA. Now, your virus is also composed of a protein shell known as a capsid. And in some viruses class, they would have a lipid envelope. Virions have full capacity for replication class when a susceptible target cell is encountered. Now let's talk about their parts class. So we have here your capsid and capsomeres. Now this is the protein coat that would surround the viral nucleic acid. This is composed of repeating mid-29 subunits called capsomeres. Generally, capsids have either helical or icosahedral symmetry. So take note of that. Klasa yung capsid nyo, this is the protein coat that would surround the viral nucleic acid. And they have two possible shapes. Pwedeng helical, katulad nito, or pwede siyang icosahedral. Then we have your nucleocapsid. This is the complete protein nucleic acid complex. Another term class, we have your satellite or defective virus. The satellite or defective virus class, these are viruses that would require a second virus, also known as a helper virus for replication. The best and most common example that of this class is your hepatitis D virus, also known as your hepatitis delta virus. It would require the presence of hepatitis B virus to complete its replication cycle. The helper virus here is hepatitis B. Ganito kasi ang class. For your hepatitis D class, for hep D to occur in a person, a active hep B infection must be present first. Must be present. That is why, class, if you are positive for hepatitis B and you do not undergo treatment, there's a possibility that hepatitis D or hepatitis delta infection can occur. So let me repeat, class, for hepatitis D to occur in a person, a active hepatitis B infection must be present. So that's a satellite or defective virus. It basically would require a helper virus. Then we have your viroids class. Now, viroids class are the smallest known autonomously replicating molecules. They would consist of a single-stranded circular RNA that are 240 to 375 residues in length and are considered plant pathogens. So viroids would only affect plants class. So may mga viruses then that would infect plants known as your viroids. Then we have your prions class. Now prions class are technically not viruses but are often we discuss in this category. Now, prions are considered infectious protein molecules class. So, hindi talaga sila considered viruses. They are infectious protein molecules that contain no definable nucleic acid and are responsible for the transmissible and familial spongiform encephalopathies. To simplify class, pag sinabi mong familial, they are typically inherited na mamana sila. Then encephalopathies are related to the brain. So these are inherited brain disorders. They would affect the brain, such as your Creutzfeldt-Jakob disease, your Kuro, your fatal familial insomnia, your Gerstmann-Straussler-Schenker syndrome, and your bovine spongiform encephalopathy, also known as your mad cow disease. Now, the pathogenic prion protein class is named as PRPSC. So PRPSC class, that is your pathogenic prion protein. This is formed from a normal human protein, PRPC, through post-translational processing. Di ba class ang in your central dogma, we have your DNA replication, we have your DNA to RNA transcription, then we have your RNA to protein translation. Now, in certain cases wherein there is an abnormal translation, what happens that instead of having a normal PRPC, which is the normal human protein, this would become your abnormal or your pathogenic prion, PRPSC. So this is related to certain mutational errors class in translation. Kaya nabubuo yung proteins na yan. Then we have the term viruses class. So these are intracellular organisms unable to reproduce itself outside the host cell. So ang viruses nyo class, to simplify, they would require a host cell. Without a host cell class, they cannot reproduce. Now, viruses could either be a haploid, meaning they only have one copy of their gene, except, may exception dito class. The only exception is that the retroviruses class. Retroviruses are considered diploid. Yung mga HIV nyo class, they are considered diploid. Now, the problem with your viruses class is that they would pass through filters. Size ranges from 20 to 200 nanometer. Very small class of viruses nyo. They only have a size of 20 to 200 nanometer. That's why they could pass through your HEPA filter of biosafety cabinets. That's why if you're going to handle viruses, BSL-3 is not enough. You need to use BSL-4, wherein you would wear a positive pressure suit. Hindi ka pwede mag-inhale ng external air when you process viruses class kasi they are not filtered. Lahat ng hangin, all of the air in a biosafety level 4 is forced outside, and you would have your own air supply or your own oxygen supply. Pag nakakita ka ng biosafety level 4 class, you would look like an astronaut or a diver, a deep sea diver. Genetic material is either DNA or RNA, and they are not sensitive to antibiotics. So they are not destroyed by antibiotics class. Instead, you would use antivirals. Now, let's talk about the viral structure class. Now, viral structure class, we have your virion. Now, virion is the complete infectious viral particle. Then we have your genome or your nucleic acid core. Now, this would contain the genetic material, either RNA or DNA, which can be single or double-stranded. Then we have your capsid, which would surround the genome. Now, your capsid class could come into different forms. As I mentioned, it could be helical or rod-shaped, icosahedral or cube-like, or complex. Pag sinabi mong complex, they have no fixed shape. They are not helical, they are not icosahedral. An example of this is your pox viridae. So this is an example because of a helical virus, your TMV. They are rod-shaped. Then we have your polyhedral, your adenovirus, spherical for your influenza, and yung complex, which is your bacteriophages. Then we have your outer membrane. This is envelope. Now, take note, class, that not all viruses have an outer membrane or an envelope. So if it's an envelope, if a virus is with envelope, it has outer membrane. It is ether sensitive or labile. So let me repeat. If a virus has an outer membrane or envelope, it is ether sensitive or labile. If it's naked, when you say naked, it's also a term used for describing non-envelope, meaning it's ether resistant and or ether stable. Now, take note of the modern classification of viruses class. So you would classify viruses in the following, 'di ba? If you classify bacteria, you could classify them based on their shape, kung koksay ba sila, kung spherical ba sila, or rod shape sila, bacilli, ganon, or spirochete sila or spiral. Now, you could classify viruses based on the following. The type of viral nucleic acid. So, is the virus a DNA virus or an RNA virus? Are they single-stranded or are they double-stranded? So, are they ssDNA or dsDNA? Are they ssRNA or dsRNA? SS would stand for single stranded, DS would stand for double stranded. Then we have your capsid symmetry. Is it icosahedral? Is it helical? Or is the virus complex? Then is it naked or none or has an envelope? Then we also have to consider structure class. So ito yung old classification nila ng viruses. Somewhat similar din naman, class. Is it DNA or RNA? Single or double-stranded? Is it helical or icosahedral yung capsid? Does it have an envelope? Something like that. And you also have to consider, class, the virion assembly. When you say the site of virion assembly, where does it reproduce? Is it the nucleus or the cytoplasm? So do remember all of this, class, kasi in the coming lessons, each virus has their own category or classification. Some viruses would be helical, some viruses would be single-stranded DNA, some would be double-stranded RNA, and so on. So this is the appearance of your virus, class. So this is your envelope. So this is an envelope virus. This is a double. Well, I believe this is a single-stranded DNA virus. Then the capsid is cuboidal. Then we also have your matrix. Then these are the different DNA viruses that we will tackle, class. So let's divide. So DNA viruses are divided based on their strand, double-stranded and single-stranded. Now let's start with the double-stranded DNA, class. So we have the non-envelope and envelope. Let's go with the non-envelope. So double-stranded non-envelope DNA viruses would include adenoviridae, which would include the virus that would cause the common cold and the viral meningitis. Then we have your papilloma viridae, known to cause your warts and your cervical cancer. Then we have your envelope, double-stranded DNA. This would include your pox viridae, known to cause your smallpox, herpes viridae, known to cause your cold sores, chickenpox, shingles, and infectious mononucleosis. Then we also have your Hepadnaviridae, which would include hepatitis B viruses and liver cancer. Then we have your single-stranded non-envelope naman, class. So for single-stranded non-envelope, this would include your Parvoviridae, known to cause your fifth disease and gastroenteritis. So that's your DNA viruses. Then the one that's most abundant, class, yung RNA viruses. So we have your single-stranded RNA and double-stranded RNA. For single-stranded RNA, we have your positive strand or positive sense and is non-envelope. So non-envelope positive strand, single-stranded RNA would include your picornaviridae, known to cause common cold, polio, and Hep A. Calliciviridae, known to cause your gastroenteritis. Then for the envelope, we have your Togaviridae, known to cause your rubella encephalitis. Flaviviridae, known to cause your yellow fever, dengue, West Nile, Hep C, Zika virus. Then we have your coronaviridae, known to cause your SARS and your COVID-19. And we have your retroviridae, known to cause your AIDS. Now, for the negative strand, we have the envelope. This would include the filoviridae, which is related to Ebola and Marburg virus diseases. Bunyaviridae, hantaviruses, rhabdoviridae, rabies, orthomesoviridae, influenza, paramesoviridae, known to cause your mumps and measles, then arenaviridae, known to cause your Lassa fever. Then for the double-stranded non-envelope, we have your Rioviridae, which is known to cause gastroenteritis. So do memorize this table class. This may come out in your quiz. Now let's talk about the viral physiology. Let's start with viral replication class. So viruses can only multiply class inside a living cell. They would require a host cell to survive. The host cell must contain the required machinery to synthesize the viral proteins and nucleic acids. So take note, if you want to differentiate viruses from bacteria in terms of replication, bacteria and fungi can replicate on their own. Now, viral replication would occur in several stages, namely the first one, attachment class. So, pag attachment, the virus would become attached to the cell by specific cellular receptors. Now, these receptors class would come in different forms, such as glycoprotein, phospholipids, or glycolipids. Now, after attachment to the host cell, to the surface receptor, it would now enter the cell, most commonly via receptor-mediated endocytosis. Pag sinabi mong endocytosis class, they would enter, or they would they would open the receptors, allowing the virus to enter. This is the same process by which many hormones would enter the cell. Now, after entry, the virus would uncoat. When you say uncoat, the viral capsid would be uncoated, releasing the viral nucleic acid. Uncoating may be achieved by host or viral enzymes that would degrade the capsid. Now, once it's uncoated, class replication would occur. What happens is that the DNA or the RNA would replicate by switching the host machinery from cellular protein synthesis to viral synthesis, and viral proteins are produced. So, ang ginagawa dyan, class, yung viral nucleic acid that was released from uncoating would attach to the host nuclear material. Then it would start creating viruses. So instead yung normal na translation, normal na transcription, it would start creating copies or viral proteins. Then after that, newly synthesized viral proteins are post-transcriptionally modified and packaged into virions that can be released from the infected host cell to infect other cells. Virions are released from the cell either by lysis or budding class. In lysis, the infected cell dies and the virions are released. In budding, the virions take some of the host cell's membrane with it as it lives. This normally does not kill the infected cell. Most of the time, class, ang mode of release ng viruses nyo is always via lysis. Kaya nga kapag meron kang viral disease, you would suffer from inflammation due to the destruction of your cells. So this is the picture depicting the class. So this is an example of an influenza virus that would affect a host cell. So there would be the attachment. It would attach to the surface receptor. Then it would penetrate the cell via endocytosis. Then it would uncoat its capsid, releasing viral content. After that release, the viral nucleic acid would attach to the cell's nuclear material. There would be biosynthesis. Then there would be assembly. New phage particles are assembled, and after that there would be release either via lysis or budding. So do remember the steps of viral replication. Attachment, penetration, uncoating, biosynthesis, assembly, and release. Now let's go to specimen collection, processing, and handling class. So lab diagnosis of viral infection would require understanding the following class: the pathogenesis of the suspected agent, the stage of infection, and the age and immunocompetence of the infected individual. It is important to select the appropriate specimen, collect the specimen carefully to optimize recovery of the infectious agent, and transport the specimens as directed to so as to maintain viability and minimize overgrowth with contaminating agent. Now, when you select your specimen class, it must be collected from the target organ most closely associated with the clinical symptoms. So kung ang suspected virus mo is an influenza virus class. So you would have coughing kapag may influenza ka, 'di ba? May cough ka, may sneezing ka. So most likely you would get respiratory samples. So you could get a swab, you could swab the patient, you could get sputum. So as much as possible, if you're going to collect samples for viral testing, you need to make sure that the specimen is collected from the organ closely associated with the symptom. Do remember that. Now, what are the reagents or materials used? The most common reagent used class in viral specimen collection is your VTM or your viral transport media. During the COVID pandemic class, very common nyo yung nakikita. Pagkatapos kayong iswab ng medtech, if you were swab, they would put the swab in a VTM. Now, VTM prevents the specimen from dying. It maintains viral viability and retards or prevents the growth of microbial contaminant. Now, VTM would contain gelatin and antimicrobial agents in a buffered salt solution. Then take note of the supplies used. So you're required class to use sterile, leak-proof screw cap containers for urine cups, disposable centrifuge 15 and 50 ml. Then you could also use smaller tubes 4 ml nunk vials suitable for holding 1 to 2 ml of BTM. Sterile cotton, dacron, or rayon tip swabs with plastic or aluminum caps. Small tip flexible swabs are used for certain samples such as urethral swabs. Do not use calcium alginate swabs or wooden shafted swabs class because yung calcium alginate swabs they would kill viruses. While yung wooden shafted swabs naman is prone to contamination. Then for tuberculin syringe with at least 26 or 27 gauge needle for aspirating vesicular fluid. Then if you want to collect blood for viruses, you need to use the anticoagulant ACD. ACD stands for acid citrate dextrose. Then for the equipment, you could use refrigerator set at 2 to 8 degrees centigrade. Now, for the collection guidelines class, take note of this. Collect specimens as soon as possible class after the onset of symptoms. Yet the chance of patient recovery is best during the first 3 days of onset and is greatly reduced beyond 5 days with many viruses. So, yung pinaka-best time class, the best time to collect viral specimens is 3 days from onset of symptoms. Example, kasi halimbawa, today, nagkaroon ka ng COVID. Today is February 25. You have 3 days from today. So, 25, 26, 27 for to have to be the best time to collect the sample. Now, after 5 days class, the chance of recovering the virus is greatly reduced. So after 5 days. So pagdating ng mga March 1 or March 2, you would have difficulty in recovering viruses. Then autopsy samples need to be collected as soon as possible after death before tissue would start decomposing. Now, in your handouts class, there is a specimen collection guide table. Later, we will go to that slide. Now, place swabs into a tube containing a small volume of VTM and scrapings and small pieces of tissue in a tube also containing VTM or saline. So, for swabs class, sa VTM nyo siya ilalagay. For scrapings and small pieces of tissue, pwedeng sa VTM or sa saline. Place fluid and bulk specimens into a sterile leak-proof container. Then add VTM to prevent drying of tissue. Collect samples as aseptically as possible to avoid introduction of contamination that could affect culture later and make accurate diagnosis difficult. Place each specimen into a separate container labeled with the patient's name or identification number, the collection site, the date of collection, and the time of collection. So take note of that class. You need to label it with the patient's name, site of collection, date of collection, and time of collection. Obtain a complete patient history, including the date of the onset of symptom. Then for the clinical findings, take note of the following. Recent exposure history. Are there any animal or arthropod contacts or animal bites? Recent travel to areas of endemic infection. Pumunta ba yung patient nyo ng Africa class? So baka meron siyang Ebola. And or did the patient take any recent vaccines? Now, all specimens class except blood should be tightly sealed, kept cold, and sent on wet ice or a cool pack to the laboratory as soon as possible, preferably on the day of collection. Now, specimens for CMV or cytomegalovirus isolation should be sent as soon as possible to the laboratory since this virus is very heat labile. When you say heat labile class, it can easily destroy by heat, and the probability of isolating it decreases with time. It is absolutely necessary that all specimens except blood and those for CMV, RSV stands for respiratory syncytial viruses. And yung VC nyo would stand for varicella zoster. Should be frozen at negative 50 degrees or below and sent to the laboratory on dry ice. Take note of that, class. So this is the specimen collection table and specimen collection and patient preparation table. So let's discuss them one by one. So for CSF class, optimum collection schedule is within 7 days of onset. Volume is 1 to 2 ml. For amniotic fluid, 3 ml. For vitreous fluid found in the eye, 1 ml. Urine should be collected within 2 weeks of onset, at least 5 ml. For blood, 5 to 7 ml in yellow ACD tube. For bone marrow aspirate, 2 to 5 ml in yellow ACD tube. For bronchoalveolar lavage, 3 to 5 ml. For nasal throat, NPS, vesical lesion, rectal eye, and conjunctival swabs for the nasal swabs class within 5 days. For vesicular and genital swabs as early as possible. Then swab in M4 tube, in viral transplant media or M4. Then aspirates, nasal tracheal sinuses should be at least 1 to 2 ml. Nasal wash within 5 days of onset, 3 to 5 ml. Feces should be collected within 2 days of onset. Amount is at least 5 ml. In container without preservative. Tissue volume is variable class. Placed in a sterile, saline, or viral transport media. Now, there's also, you have to consider also class patient preparation. So you would have, for truth wash, have the patient gargle with 10 ml of sterile balanced salt solution or triplicase soy broth expelled in a sterile container. Then for urine, random clean cut sample could be collected in a sterile container. For blood, 7 to 10 ml of blood in a yellow ECD tube. Reject blood more than 72 hours old from the time of collection to the time of processing. For nasopharyngeal swabs or your NPS class, insert a flexible fine shafted swab through the nostril into the nasopharynx and rotate swab gently a few times. Place into M4 viral transport media. For the vesical fluid, withdraw vesical fluid using a tuberculin syringe or swab the vesicle and place it in an M4 culture transfer tube or 1 ml of BSS or TSB. Reject crusted dry lesions are not vesicles and are not appropriate for virus isolation. For tissue, collect aseptically and keep moist by placing in sterile container with 2 ml of BSS or your Hanks phosphate buffer saline or TSB saline or viral transport media M4. Then for stool, approximately 5 milligram stool placed in a tightly capped sterile container without preservatives. A swab covered with stool is acceptable for culture but not preferred. Swabs are unacceptable for Clostridium difficile toxin. So do memorize the tables class. They may come out in your exam and quiz. Now let's have a general overview class of the lab tests for the identification of viruses. So we have your direct detection, we have your indirect examination, and your serology. So sa serology class, you would be able to relate to this in your immunosero because in identifying viruses, there is what we call the presence of antigens and nucleic acids and even antibodies. Now, in direct examination, the clinical specimen is examined directly. In the indirect examination, the specimen is placed into a cell culture, eggs, or animals in attempt to grow the virus. This is called virus isolation. Then serology would actually constitutes by far the bulk of the work of any virology lab. So most of the laboratory class that would handle viruses, the main type of test they would do is more on serological tests. A serological diagnosis can be made by the detection of rising titers of antibody between acute and convalescent stages of infection or the detection of IgM. In general, the majority of common viral infections can be diagnosed by serology, and the specimens used for direction detection and virus isolation is very important. A positive result from the site of disease would be of much greater diagnostic significance from those for other sites. So, to give you an example, class, if a patient is positive for herpes simplex encephalitis, a positive result from the CSF or the brain is more significant than a positive result from an oral ulcer, since reactivation of oral herpes is common during times of stress. Yung herpes virus nyo kasi class, to give you an idea, your herpes virus is capable of going into a latent form. Pag sinabi mong latent form class, this is a dormant form. Pag dormant, hindi siya nakakakos ng sakit, it's asymptomatic. Now, the problem with herpes, if a person who has latent or dormant herpes suffers from extreme stress or would encounter another type of disease that would affect his or her immune system, the herpes would become active. It would become active. And in some cases, one form of active herpes would cause oral ulcer. Now, the problem with having an activated oral ulcer class is that malilito yung doctor if the patient is having a new or recent herpes or if it's an old. That's why it's very important class if a patient has encephalitis due to herpes, a positive CSF would be greater significance kasi it helps the diagnostician identify that the disease is a new case, not an old one. Now let's talk about the direct examination of specimen. So for direct examination of specimen class, you would use your EM microscopy and also immune electron microscopy. Then you could also use light microscopy, looking for histological abnormalities, such as your inclusion bodies. Then antigen detection using immunofluorescence, ELISA. Molecular techniques for the direct detection of viral genomes. Then we have your indirect examination. We have your cell culture, cytopathic effect, heme absorption, confirmation by neutralization, interference, immunofluorescence. Egg pox on chorioallantoic membrane or your CAM, capable of producing heme agglutination and inclusion bodies. Animal disease or death confirmation by neutralization. Then we have your serology, detection of rising titers of antibody between acute and convalescent stages of infection, or the detection of IgM in primary infection. So there are classical techniques and newer techniques class used for serology. So the classical techniques, we have your complement fixation tests, your hemagglutination inhibition tests, your immunofluorescence technique, your neutralization tests, and your single radial hemolysis. Now for the newer techniques, we have your RIA or your radioimmunoassay, your EIA or your enzyme link immunosorbent assay, particle agglutination, Western blot, recombinant immunoblot assay or your RIBA, and your line immunoassay. Now let's go with the direct examination, or let's discuss the direct examination of specimen. Now the direct examination of specimen class is also called a rapid diagnostic method. They would give a result either within the same or the next day. This is extremely useful in cases when the clinical management of the patient depends greatly on the rapid availability of lab result, such as the diagnosis of RSV infection in neonates or severe CMV infections in immunocompromised patients. Now, it is very important, class, to take note that not all direct exam methods are rapid, and conversely, virus isolation and serologic methods may sometimes give a rapid result. Now, in today's laboratory class, most tests are becoming rapid diagnostic methods. They are starting to play an increasingly important role in viral infection. So let's start with the first type of direct examination, yung antigen detection. Now, this would include immunofluorescence testing for viruses such as your RSV, influenza A, influenza V, and adenoviruses. Detection of rotavirus antigen in feces, the PP65 protein in for CMV antigenemia tests, the detection of herpes simplex virus and varicella zoster virus in skin scrapings, and the detection of hepatitis B surface antigen in serum. Now, the main advantage of this assay is that they are rapid to perform, with the result being available within a few hours. So, ang pinaka-familiar kayo dito, class, itong HBsAg, di ba? You will be required to take this bago kayo mag-intern. So, dapat mag-negative kayo dyan. Then, what are the disadvantages of antigen detection, class? So, the technique is often tedious, time-consuming, result is difficult to read and interpret, and the sensitivity and specificity of the is poor. Quality of the specimen obtained is of utmost importance in order for the test to work properly. So these are examples of for antigen detection. We have your herpes simplex virus infected epithelial cell from a skin lesion. Then we have your CMV PP65 antigens detected in the nuclei of peripheral blood neutrophils. Now let's go to another method, yung electron microscopy, class. So virus particles are detected and identified on the basis of morphology. A magnification of around 50,000 times is normally used. EM now is mainly used for the diagnosis of viral gastroenteritis by detecting viruses in feces, such as rotavirus, adenovirus, astrovirus, calicivirus, and Norwalk-like viruses. Occasionally, it may be used for the detection of viruses in vesicles and other skin lesions, such as herpes virus and papilloma viruses. The sensitivity and specificity of EM may be enhanced class by immune electron microscopy, whereby virus specific antibody is used to agglutinate virus particles together, thus making them easier to recognize or to capture virus particles onto the EM grid. The main problem with EM class is that it's very expensive. Milyon ang gagastusin mo if you want to buy an EM. Uh, In addition, the sensitivity of EM is often poor, with at least 105 to 106 virus particles per ml in the sample required for visualization. Therefore, class, you need to have a very trained observer. With the availability of reliable antigen detection and molecular methods, EM is becoming less and less widely used. So this is how you would use your EM class. So you would view viruses and you would describe them or identify them based on their morphology. So from left to right, we have your rotavirus, we have your adenovirus, we have your astrovirus, then we have your Norwalk-like viruses. Then you could also use light microscopy class. So replicating virus would often produce histological changes. So kapag nagre-replicate ang viruses nyo, it would cause changes in the tissue. These changes may be characteristic or non-specific. An example of these changes class is the formation of viral inclusion bodies that are either found in the nucleus or cytoplasm. An example of a common inclusion body class is yung tinatawag nilang negri bodies. Negri bodies class are found in rabies virus infected individuals. Cytomegalic inclusions found in CMV infections. Although not sensitive or specific, histology nevertheless serves as a useful adjunct in the diagnosis of certain viral infection. Then we have your viral genome detection. So this time, class, you're going to use molecular methods, which would use the detection of the viral genome. This is the future of viral diagnosis, class. So classical molecular techniques such as your dot blot and southern blot would depend the use of specific DNA or RNA probes. So meron kasing yung southern blot class. Southern blot would be used for DNA. Northern blot would be used for your RNA, and yung western would be used for proteins. So take note of that. Now, newer molecular techniques would include your PCR, LCR, NASBA or nucleic acid based amplification, and yung branch DNA assay. Now, all of this would depend on some form of amplification. Either they would target the nucleic acid or the signal itself. Branch DNA is essentially a conventional hybridization technique with increased sensitivity. However, it is not sensitive as PCR and other amplification techniques. So the one you're most familiar class dito is yung PCR. This is the only amplification technique which is commonly used. PCR is an extremely sensitive technique. It is possible to achieve a sensitivity of down to 1 DNA molecule in a clinical specimen. However, PCR has many problems. Ang pinaka-common is contamination, since only a small amount of contamination is needed to give a false positive result. In addition, PCR is so sensitive compared to other techniques that a positive PCR result is often very difficult to interpret, as it does not necessarily indicate the presence of disease. This problem is particularly great in the case of latent viruses or latent viruses such as CMV. since latency MV genomes may be amplified from the blood of healthy individuals. My case kasi class were in, if you still remember, during the pandemic, let's say you're already, you're recovered. May mga individuals kasi that they have recovered from COVID 4 months na. Buwan na. Siguro nagka-COVID sila ng January. Tinest sila ng May. Positive pa rin sila sa COVID. The reason for that class that even dead viruses in the body are being detected by your PCR. Ganun siya ka-sensitive class that even dead viruses in a healthy individual are detected, causing false positive results. Now, despite all of this, class PCR is being increasingly used for the rapid diagnosis. This is especially as the cost of the assay would come down and the availability of close automated systems are created, such as yung real-time PCR nyo and other techniques, such as your LCR and NASPA, are also susceptible to contamination. But this is decreased class due to having a closed system. Sa PCR kasi class, the problem with your PCR is that you would use different areas. Pupunta ka sa iba't ibang location and you would prepare the reagents in different places. Now let's go to the second method class. So we're done with your direct examination. Let's go to your indirect one or your virus isolation. So cell cultures, eggs, and animals may be used for isolation. However, eggs and animals are difficult to handle and most viral diagnostic labs would depend on cell culture only. Now, there are three types of cell culture. We have your primary cell, semi-continuous cell, and continuous cells. Now, primary cells would include monkey kidney cells. These are essentially normal cells obtained from a freshly killed adult animal. These cells can only be passaged once or twice. Then for semi-continuous cells, this would include your human embryonic kidney and skin fibroblasts. These are cells taken from embryonic tissue and may be passed or used up to 50 times. Then we have your continuous cells such as yung HeLa, Vero, Hep2, LLCMK2, and yung BGM. These are immortalized cells, usually tumor cell lines and maybe passage indefinitely. Ang pinakakilala dyan class si HeLa. Siya yung source ng cell line na yan. Now, primary cell culture is widely acknowledged as the best cell culture system available since they support the widest range of viruses. However, they are considered expensive class. During my time when I was a student, we had a one group during the thesis sa research namin class. Meron kaming isang group na nag-undergo ng thesis experiment wherein they use cell lines. Ang isang cell line class, if I recall, nagka-cost ng around 120,000 pesos. So it was a very expensive research what they did. Now, continuous cell lines class are the easiest to handle, but the range of viruses supported is often limited. Take note of that. So you would also identify growing viruses class. So when your viruses grow class, they would usually produce a so-called cytopathic effect. Now, cytopathic effect may be specific or non-specific. Herpes simplex virus and cytomegalovirus would produce a specific cytopathic effect, whereas enteroviruses do not. Then they're also capable, some viruses are capable of heme adsorption, wherein cells acquire the ability to stick to mammalian red blood cell. Heme absorption is mainly used for the detection of influenza and para-influenza viruses. Confirmation of the identity of the virus may be carried out using neutralization, heme absorption inhibition, immunofluorescence, or molecular tests. So these are the different cytopathic effect class. So for herpes simplex and enterovirus, The cytopathic effect seen is the ballooning of cells. So, nag-enlarge yung mga cells that would be affected. Now, for RSV, there is the Cincha formation class. Yung nagkukumpol-kumpol sila. So, that's the cytopathic effect. Then, we also have problems with cell culture. So, the problem with cell culture class is having a long period of culture up to 4 weeks before a result is available. Also, the sensitivity is often poor and would depend on many factors such as the condition of the specimen and the condition of the cell sheet. Cell cultures are also very susceptible to bacterial contamination and toxic substances in the specimen. Lastly, many viruses would not grow in cell culture at all. Such viruses would include Hep B and C, diarrheal viruses, and parvoviruses. So yung mga yan class, they do not grow in a cell line. Then we have your rapid culture techniques, wherein viral antigens are detected 2 to 4 days after inoculation. Example of rapid culture techniques would include shell vial cultures and your CMV DAF test. In the CMV DAF test, the cell sheet is grown on an individual cover slip in a plastic bottle. After inoculation, the bottle is then spun at a low speed for 1 hour. This would speed up the absorption of the virus. And then incubated for 2 to 4 days, the cover slip is then taken out and examined for the presence of CMV, early antigens by immunofluorescence. The role of cell culture, both conventional and rapid technique in the diagnosis of viral infection, is being increasingly challenged by rapid diagnostic methods, such as your antigen detection and molecular method. Therefore, the role of cell culture is expected to decline in the future and is likely to be restricted to large central laboratories. So this is an example, the same absorption of RBC on the surface of a cell sheet infected by mumps virus. Now let's talk about the pathogenesis of certain viruses. So as with all other infectious agents which would cause human disease, the outcome of the interaction of a particular virus with the human host is dependent on pathogen and host factors. Now, viral strains within the genus may have differential cell tropisms, replication capacities, and cytopathogenic effect. Pag sinabi mong cell tropism class, what type of cell would be affected? Then what are the replication capacities? Do they replicate fast? Do they replicate slow? Then what are the effects? Yung mga cytopathogenic effects nila. What happens to the cell? What happens to the tissues? An example class, strains of HIV may target monocytes, macrophage, or T lymphocytes. Ang HIV nyo class, they target T cells. Kaya most of the time kapag ang tao may AIDS, they would have low T cell count. Now they may use 5 different co-receptors or chemokine receptors, yung CCR5 or yung CXCR4 on the cell surface. They may replicate to different levels and may induce different degree of cell killing. These traits have direct clinical correlates for HIV-infected persons with respect to the rate of CD4 cell decline and progression to clinical AIDS. On the host side, the nature of the exposure and the host immune status are probably the two most important determinants of outcome. So, take note, class, for a person to be infected, you have to consider the nature of the exposure. Pag sinabi mong nature of the exposure, is it directly affecting the individual to the point na madali siya magkasakit? And you would also have to consider the immune status of the individual. What are the key elements to the virus-host interaction? So you would consider the viral strain, inoculum size, route of exposure, susceptibility of hosts. Is the patient vaccinated? Did he have a natural immunity from a past exposure? Immune status and age of hosts. Now, the net result, so yung interaction between host and virus could lead to the following class. No infection, abortive infection with limited viral replication, asymptomatic infection, symptomatic infection. Then, depending on the agent and the immune status of the host, there could be a persistent, latent, or self limited infection. So these are the pathogenic steps in a human infection class. So depending upon the agent, the virus would enter to the skin, the mucous membrane, the respiratory tract, gastrointestinal tract via transfusion or transplanted organ or via maternal fetal transmission. Nowadays, class, ang pinaka-common virus that is being transmitted in the Philippines is HIV via parenteral route, yung exchange of body fluids, reuse of needles. So yun, that's how you would get certain viruses. Then there would be local replication class at the site of the inoculation. So from the site of entry, viruses would start replicating. Now, certain viruses would exhibit pathology at the skin or mucous membrane, such as herpes simplex virus or human papilloma virus. Pag sinabi mong exhibit pathology at the skin or mucous membrane class, there would be different rashes, there would be different skin disease that would form, such as in the case of herpes. Pag may herpes ka, class, magkakaroon ka ng skin ulcer. Or pag may human papilloma virus ka, this is known to cause your warts. Now, for some neurotropic viruses that may spread along the nerve route to the ganglia, such as HSV, or the CNS, such as your rabies virus, for other neurotropic agents, the CNS is seeded following viremia. So may mga viruses class that they would go directly to the nerves, such as your herpes simplex and yung rabies virus. Kaya nga nakakatakot pag nagkagat ka ng aso, you need to make sure that you would get vaccinated immediately. Now, for many agents, there is replication in the regional lymph node with subsequent viremia and spread to target organs. Some viruses would travel in the bloodstream, in the plasma, such as your picorna viruses. Others are would target cells, such as your cytomegalovirus. Replication in target organs may lead to local damage and further rounds of viremia. An example yung nagre-replicate na sa organ class in the case of hepatitis B. In hepatitis B class, in advanced cases, it would affect the liver, leading to jaundice, leading to cirrhosis. Non-specific and specific host immune response would come into play to try to control and downregulate the viral replicative process. So to give you a better explanation class, so the virus would enter the human causing a primary infection. So it could cause respiratory or would target respiratory, gastrointestinal, urogenital organ. Then it would lead to viremia. Viremia is the presence of virus in the blood. From the site of entry, it would now go to the organs, to the nerves. Then it would replicate or localize in the lymph nodes, leading to a secondary infection, wherein it would start migrating to different organs, leading to the development or the signs of diseases. Now, what are the terms used in describing an infection of an organism? So do memorize these terms, class. So we have your lytic infection. So when virus enters the cell and hijacks its cellular machinery to rapidly multiply and in the process kills the cells is termed as lytic infection. Many influenza viruses, another term yung lysogenic infection. It is the process characterized by the incorporation of viral DNA to the cellular DNA. Once incorporated, the viral DNA would replicate along with the host DNA. The incorporated viral DNA permits the host cell to undergo normal cell cycle. Then we have your acute infection. It is a rapid onset of disease symptoms resulting in severe illness or death of the infected animal. Then for chronic infection, it is a prolonged infection in which the organism is not immediately killed and may carry the virus for a long period of time. Now, what are the different terms used to describe virus transmission? We have your horizontal transmission. This is defined as the transmission of virus or other pathogen to hosts at any age after birth. So pag horizontal, from the line horizontal kas ganon, meaning hindi siya namamana. You could get the disease at any point of your life from other sources. Then we have your vertical transmission. This is the passage of a virus from mother to the newborn child. Then we have your zoonosis. This is defined as the disease which is naturally transmitted between animals and man, such as your rabies, H1N1 virus, Rift Valley fever virus. So ang zoonosis nyo or zoonotic diseases nyo class are usually diseases only in animals, but there are certain cases wherein humans can also acquire them. Sometimes the virus can be transmitted through an vector or insect vector, such as your arboviruses. Viruses present in the saliva of the infected insect are transmitted during feeding of blood meal to the susceptible hosts. Then we have your persistent infection. This is a condition where the virus would remain associated with the cell without actively multiplying or killing it. This often occurs when the viral genome gets integrated into the host genome, seen in retroviruses, and sometimes without integration, seen in herpes viruses. Now, persistence can be categorized into three types. Virus genome would persist within the cell without actual release of the virus. So, di ba yung viruses nyo class, once they enter the cell, they would usually lyse the cell or they would bud. But there are some viruses such as your retroviruses wherein they would just stay within the cell. Then viruses would be released sporadically but remain in a state of latency for most of the time, such as your herpes simplex virus. So sa herpes simplex virus kasi class, the virus would become dormant, as I mentioned earlier, would become dormant in some cells. Then viruses release continuously without lysis of the host cell, seen in hepatitis B virus. Then we have your multiplicity of infection or your MOI. This is the ratio of the total virus infected to the number of target cells in an infection condition. This is usually used to describe the infection of a cell type grown in vitro in a culture system. So we have your infectious dose 50 or ID50. This is the dose required to infect 50% of inoculated animals. Lethal dose 50, the dose required to kill 50% of the inoculated animal. Then incubation period, this is the time between the initial infection to the actual onset of disease symptoms. This could range from a few days to years. Kaya nga class, yung incubation period is also known as your window period. Let's say you had a sexual intercourse with an HIV positive individual. At day 1 up to the third month, let's say na-acquire mo after having sexual intercourse. From day 1 up to the third month, no symptoms would show. So yung incubation period niya or yung window period niya is up to 3 months. Walang symptoms class. But you already have the virus in your body. That's why nakakatakot if you're the type of individual that has multiple partners, you could easily acquire HIV nowadays because of the change in our society. And the terrible part is that this virus has a window period wherein you will not be able to know immediately if you have the virus. So this is an example of a diagram class showing the difference between the three infections, acute, latent, and chronic. So if you take a look, pag acute class, increase infection in the early years. Pag latent naman siya class, increase infection in the early years with a possibility of reactivation in the future. So pag latent, from having a high infection at the early years, it would go dormant or not detected siya sa body mo. Later on, in a certain part of your life, it could become reactivated, causing a new infection. Then it would become dormant again, and after a few months, after a few years, magre-reactivate na naman siya. Kapag chronic infection naman, decrease infection in the early years, increase or constant infection in the future. So the problem with chronic infections class is that they do not subside. They would tend to have a constant infection even up to the future. So from a high infection, magdi-decrease siya, but not to the point that it would subside or would be totally gone, but it would lead to a constant infection in the future. Now let's go to the last part, the immune response to viral infection. So we have your non-specific immunity. This would refer to the elements of the immune system that can clear virus or virus-infected cells immediately upon or shortly after viral exposure and are not dependent upon immunologic memory. Example of non-specific immunity are your WBCs, such as your phagocytic cells, your neutrophils, monocyte, and macrophages. Cytokines, such as your interferons and chemokines. Natural killer cells, and some antivirals found in your body. Then we have your specific immunity. So this refers to antigen-specific B responses that lead to the development of antibodies, cytotoxic T cell, and antibody-dependent cellular cytotoxicity, or your ADCC. In some instances, class, an intense immunologic reaction to a viral agent can result in immunopathology and a serious clinical syndrome. A prime example is dengue, which is likely due to antibody dependent enhancement and T cell activation on re exposure to dengue virus. So, these are some examples of your innate and adaptive immunity. Here, you would find your interferon class, which would help fight the viruses. Then we have here your B cells. B cells would become plasma cells that would create antibodies. And then we have your NK cell, or your natural killer cell, which would help in killing infected cell. Then we have your cytotoxic T cell. Cytotoxic T cell, or your CD8 positive T cell, which are known to kill also infected cells. So that ends the introduction to virology. Thank you.